Hi everyone, I'm Maggie, a graduating PhD student from UCLA. I was previously an ALS doctoral fellow. I would like to share with you today our work strain-mediated multifluoric systems for life science applications, facilitated by XMCD and XMCD PIM, followed by a study on localized strain profile generated by surface electrodes using X-ray microdiffraction. I've been a user of multiple ALS beam lines to investigate material properties of our composite multifluoric systems, focusing on both magnetism and strain. So for the first part of the talk, we'll look at the strain-mediated multifluorics and microfluidics platform. Such platform can be used for life science applications where single-cell manipulation is crucial. And one of the motivating applications is personalized cell therapy. For example, T cells are critical to our immune system. CAR T cells with special receptors can be used to recognize and attach to tumor cells. And one of the key studies is to observe the cell behavior over time, as well as identifying the most effective cells. This is a domain where we believe our platform can play a role. In this overview conceptual image here, you see we can culture the cells while having them trapped to circular magnetic disks and selectively release the ones by tuning the strain. So before diving in the platform, I will give you a quick overview of the cell sorting state-of-the-art. On the horizontal axis, we have throughput, and on the vertical axis, we have information content per cell. From the bottom up, if we only care about morphology, such as cell shape, acoustic and dielectrophoretic sorting would work. Next up, we have magnetic activated cell sorting techniques, which attaches superparamagnetic beads to the cell. It has high throughput, but one cannot distinguish cells based on their expression level. So this is more of a binary response that we are looking at. For the single cell resolution, fluorescence-assisted cell sorting is prevalently used. On the top level, the methods have temporal data available, meaning that one can measure over time the behavior of cells. And Berkeley Light uses optoelectronic tweezers with projected light to manipulate single cells and particles. Our approach will use magnetic stray fields to control the cells, microfluidics flow, and strain to release the cells. So the diagram here first illustrates coupling between primary ferroic orders, ferromagnetism, ferroelectricity, and ferroelasticity. Multifluoric materials exhibit more than one of these ferroic orders in the same phase. And magnetoelectric multifluorics that exhibit both ferromagnetism and ferroelectricity are of particular interest. They have been used in sensors, actuators, memory devices, RF micro devices, etc. And our work focuses on composite multifluoric heterostructures, Terfenol-D on PMMPT. Fundamentally speaking, the magnetization in Terfenol-D can be controlled by the induced strain from ferroelectric PMMPT via the converse magnetoelectric effect. So our work combines a multifluoric platform with microfluidics channels to achieve magnetic single cell separation. The idea here is to show reliable magnetic bead capture by magnetic disks with no external magnetic field bias, and then work on cell capture by the same disks, and finally cell release by controlling the strain coupled to the magnetic disks. We would apply electric field to the ferroelectric substrate PMMPT to generate a strain and to perturb the existing magnetic states. And that would reorient the magnetic domain and subsequently releasing the attached cell. So first of all, we need to check the magnetic domain states in Terfenol-D microdisks. XMCD is used to image the in-plane magnetization of Terfenol-D disks that are 20 micron in diameter. Here we went to iron L3 and L2 absorption edges and took images. Since at iron L3 and L2 absorption edges, the XMCD has opposite signs of magnetic dichroism. The opposite contrast in these two pin images confirm the observed image contrast is indeed magnetic related, and the structure exhibits a net single domain configuration. Well, compared to other soft magnetoelastic materials such as iron Fe-B, iron gallium, the sputtered polycrystalline Terfenol-D has anonymously large single domain structures, an order of magnitude larger in dimension than previously thought possible. More importantly, for the targeted application, the magnetic domain sizes are on par with the single human sizes, around 10 micron plus. This large single domain magnetoelastic structure paves way to a new scale of micromagnetic capture large enough to localize and capture a single cell. So here on the bottom right, we show a magnetically tagged cell being captured by the initialized magnetic stray field from a single disk. Next, we will apply electric field to release the cell. Well, after observing robust capturing behavior from the Terfenol-D microdisks, we bonded microfluidics channel to the multifluidic device to introduce fluorescently labeled magnetic beads. We see the beads being captured on the magnetized poles of the single domain disks. And when we apply a voltage of 400 volts to the top and bottom surface of 011 PMMPT, it could generate strain up to 2150 ppm. The magnetic domain would reorient due to this electrically induced strain, which induces this easy axis along the 01 - 1 direction for the single domain. Consequently, we could see the bead move to a new location aligned with this new easy axis. And for a multi-domain microstructure, the net effect is negligible. So next, we would attach magnetic beads to single cells and track the cell behavior. This video here shows strain-induced bead and cell release from the Terfenol-D disks, where we would place the bead at a different orientation compared to the flow direction, and see how the bead as well as the cell would release. So here in the bottom right figure, individually magnetically labeled single cells can be released from the Terfenol-D to the outlet of the microchannel. When the electric field is applied and sufficient differential strain is introduced, the X component of the magnetic stray field would decline, and it's no longer in balance with the drag force along the flow direction. Then the cell could be released. The cells that are trapped in the device are viable for cell culture and available for downstream applications. Well, to sum up, multifluidics present an opportunity to automate single cell selection based on functional and time-dependent properties. And it can potentially addressing those fundamental challenges for biotechnology and development of optimal cell therapies. And such array addressable control of magnetic components could also be used in other practical applications, such as magnetoelectric storage devices, magnetic sensors, and voltage tunable microwave devices. And to achieve more robust element control element level control of individual magnetic microdisks, we would then look into the capability of generating diverse strain profiles across a single ferroelectric substrate. I would like to thank my colleagues and mentors for making this project possible. And coming up, I'll be discussing a study of element level control of magnetic arrays using surface electrode array. So to get a more versatile strain profile for element-wise control of micromagnets, we will look into use the surface electrode arrays to generate localized strain profiles across a single substrate. And surface strain mapping is used for us to examine optimal electrode design. And this work would serve as a roadmap or reference for designing compact programmable electrode arrays for multi-Freud applications. So the motivation of this work is to achieve element level control of magnetic arrays with strain. So we noticed that in order to get the in-plane strain to support element level control, first of all, we would prefer to have a differential in-plane strain for manipulating the magnetism. So here on the left-hand side, it's also from our work previously published in Nano Letters. We used X-ray microdiffraction to get the average strain along two principal strain directions, one along the O1 minus 1 and one along 100. So along one direction, we have tensile strain and the other compressive strain, and then we will get a differential strain, fairly large for us to control our magnetic structures being deposited, being grown on top of these PMMPD structures. Well, so these PMMPDs have O11 cuts, but if we are using those O1 cut PMMPD, with global electrodes, we will get compressive strains along both directions that we are looking at in plane O10 and 100. So these are not ideal for us to generate differential strain, but these type of substrates are useful for some actual growth. So the next part is that we want to achieve element level control, and we noticed that global electrodes would not work. So what we are looking at, so some options that have been developed by our research center is to use the surface electrodes for strain engineering. So previously, Ji Cui has proposed work of using those pattern electrodes on PCT, and this will generate in-plane differential strain. So motivated by this work, we decided to look into having arrays of surface electrodes and on one PMMPT, and then examine how how the strain profile will look like. So we'll be using the X-ray microdiffraction for this experiment, similar to what we have done earlier in Nano Letters paper. Here, a focused X-ray beam with a diameter of 1.5 micron is incident at 40 degrees on the sample surface, and is scanned along the 100 and 010 crystallographic direction of 001 cut PMMPT. The step size we use here is 10 micron, and the diffraction pattern at each step then translates to the strain information by comparing to the unstrained state of our crystal. So we can then calculate the electrically induced strain for each pixel and visualize them in 2D strain maps. Here, apart from the sample A that we mentioned in the previous slide that are covered with global electrodes, top and bottom, we also have sample B and C with patterned surface electrodes. So the differences here is among sample, we have varied separation gap between those electrode arrays, as well as varying the angle of those electrode pads. And the main difference between sample B and sample C is that C has the same gapping distance between all the electrodes, and it also has a more compact, denser electrode distribution. Okay, and then here on the right-hand side, these are the regional strain profile, and each square region here is 250 by 250 micron in size. And the experiments were being conducted on beamline 12.3.2 at ALS. So here is a sample result from one of the region in sample B. And here we see that along the x direction, we are getting a positive tensile strain. And along y direction, we are getting negative compressive strain. And then the next step is to getting the average of the strain in each location and plot it out those volume plots. And the yellow ones, they are along the x direction, and then the blue ones are along the y direction. And here we also plot the difference in those strains and treat these as a total strain. And those data are being collected at 400 volts. So one observation is that even though the averages of this so-called deviatoric strain or the strain difference are all around 1,000 ppm, we still see a wild distribution in those strain values according to those volume plots. So it does somehow imply that the distribution of those, the strain, the electrode pads, they can also influence one another. So this is something I'll be showing you later in the slides. So the take-home message of this slide is that we also look at the average strain from both experiment and simulation. And along x direction, we always get tail strain. And along y direction, we are getting compressive strain. So we are indeed getting regional differential strain, which are on the order similar to what we are seeing generated by those global electrodes on 011 cut PMMPT. So these profiles are quite similar. So that's good news. Okay, now moving forward to look at the effect of adjacent electrodes. So as I mentioned, B and C, one of the main difference is that C has more densely packed electrode arrays. And here we compare region 3 and 12 from B and C because they are identical in geometry. And we notice that here we are comparing the ratio between the strain along y direction and x direction. And we see that having these adjacent electrodes do make the local strain more anisotropic by comparing C versus B. So it's slightly larger in most cases when we look at the regions in C. Okay. Well, next part is to examine how the separation distance between the electrode array and also the angle of placement would affect the microstrain distribution. So here we show both the simulation results and experimental results. Simulations are those dotted lines, and then the experimental results are those dots, R3, R2, R1 regions that are being marked here with a circle and arrow bar. So what we see here is that separation gap does matter. And the angle that we have explored here, they are not so important in the strain distribution. So those can serve as the design guidelines for the next step, electrode spacing design. Finally, I would like to thank my colleagues and members on this project. And thank you very much. Any questions?